On a public health analyst's wall, a map told the story before anyone said a word. By July 2026, more than 1,600 people across 5 Midwestern states had reported the same unsettling illness. Along a stainless food service prep line, it looked like any other delivery. But investigators kept circling back to one thing on the plate: shredded iceberg lettuce. Before anyone could say for certain why, a supplier recalled it. In a modern lab, a molecular curve rose exactly the way investigators hoped it would. A laboratory test appeared to confirm the parasite directly in the food. Then came a strange reversal. A sealed sample sat on the table, an empty microscope field behind it. There was no confirmed positive food sample, yet the recall remained. Spread across an investigator's desk, meal notes, a receipt, supplier invoices, a shipping map. So how do investigators trace a parasite that no lab could catch red-handed? One interview, one invoice, one meal at a time. Long before this outbreak, in an early 1990s laboratory, scientists once looking at this very organism, glowing under ultraviolet light, had mistaken it for blue-green algae. The evidence was real. But what did it mean? That question would take science more than a decade to answer, and public health investigators decades more to ask all over again. In quiet kitchens and back bedrooms around the country, people began reporting a stubborn illness that would not quite go away. Nothing dramatic, just fatigue that refused to lift. Meals went unfinished. The pattern was consistent: watery diarrhea, lost appetite, and a person left deeply tired. On a real calendar, sick days marked themselves in a pattern. Some would recover, only for the symptoms to return days later, as if the illness had never actually left. By July 20th, the CDC had received reports of 4,173 laboratory-confirmed infections acquired inside the United States. Cases had been reported in 41 states. 308 people had been hospitalized. No deaths had been reported. At least 7,400 additional cases reported by state and local health departments were not yet laboratory confirmed and still required investigation and analysis. That last number needs care. It did not mean another 7,400 cases had already been confirmed. CDC said national counts could continue to rise as additional reports were investigated and confirmed. At dual monitors, an analyst worked through record after anonymized record. Each one had to be checked for duplicates, travel-related infections, and cases that fell outside the outbreak's own definition. Beside the national map, a second panel hovered, similar but not the same. There was a second headline number to keep separate from the first. More than 1,644 reported specifically within the 5-state cluster. Picture 2 data cylinders side by side, a blocked plus sign between them. These were separate datasets, built on different definitions and different dates. They should not be added together. Not every dim cluster on the map connected to lettuce at all. Some of the reports under review belong to other outbreaks and illnesses, unrelated to this one entirely. A single case moves through 3 desks before it is final: local, state, federal. Local, state, and federal numbers also differed because each counted the same case at a different stage of confirmation. Before a case counted at all, investigators had to check identity, address, and travel history to be sure it was not already counted somewhere else. To do that, investigators built something like several cameras trained on the same moment, interview after interview, all circling back to one simple question: what did you eat? A meal looks perfectly ordinary going down. Eating a contaminated meal does not make a person sick immediately. Picture a short timeline of days, each one lit in turn. Symptoms typically start around a week later, sometimes sooner, sometimes 2 weeks or more. The plate is long gone, only faint outlines remain. That delay turns an ordinary meal into a memory test days or weeks after the fact. Laid out like evidence on a table, a meal comes apart piece by piece. Investigators need every ingredient: the tortilla, the protein, the garnish, salsa, herbs, anything that touched the plate. Card by card, the desk fills up. So public health workers interview patients and compare their answers, meal by meal, looking for anything that repeats. Lay the meals side by side, and one thing keeps recurring. Different patients, different restaurants, different meals but share one ingredient, again and again. One state carried a striking share of the workload. In Michigan alone, investigators built detailed food histories from 190 people. Out of every 100 interviewed, the pattern jumped out immediately. About 90% of those patients had eaten iceberg lettuce. A magnifying glass hovers, but nothing stamps a verdict yet. A shared ingredient did not prove anything on its own, but it was a strong clue. Purchase records, invoices, a distribution map, lot stickers, so the FDA started working the paper trail backward, from restaurant to distributor to farm. Different restaurants, different cities, different paths, and yet those different paths converged on one supplier. 2 kitchens, 2 delivery doors, 2 different crates arriving. Not every location received lettuce from that same traced route, even ones that served a similar meal. Behind a plainly worded sign, cold storage workers set the recalled cases aside. On July 17th, the supplier recalled its iceberg lettuce, a professional, unhurried food safety action, not a panic. The recall was not proof of one farm or one field. It closed one branch of the investigation, leaving the wider national rise unresolved. To understand what investigators were even looking for, the story has to go back decades, to before anyone had a name for this organism, back to when scientists could see it, but could not yet explain it. In a modest tropical clinical laboratory, staff examined stool slides under period microscopes. The earliest recorded observations came from Papua New Guinea in 1977 and 1978. At his desk, he sketched 3 unfamiliar round organisms, his notes barely legible. In 1979, R.W. Washford, a parasitologist, published the first case series describing what he had found. The classification boxes on the specimen card stayed empty. He believed it was probably some kind of intestinal parasite, though it still had no modern name. Over the years that followed, it kept turning up, in travelers, in children in Peru, in people with weakened immune systems from AIDS. On a clinician's chart, the same waves kept rising and falling. The pattern read strangely familiar: watery diarrhea that could last for weeks, fade, and return. Transparent, double-walled, faintly granular inside. Under a microscope, it appeared as round bodies, about 8 to 10 micrometers across. 10 of them, laid edge to edge, would barely span the width of a fine human hair. Faint, competing sketches surrounded it, none quite settled. For years, its basic identity was unclear. Side by side under one scale, the 2 organisms looked deceptively alike. One early guess, a larger version of Cryptosporidium, a related parasite already known to science. But the evidence kept pointing somewhere else entirely, toward a completely different kind of organism, unexpected for an intestinal illness. Vivid blue against total black, the interior darker still. Under ultraviolet light, the outer wall glowed. That same glow could look blue or green, depending on which filter was used. Standard stains behaved strangely too. Some took the stain only partly, others pale, all the same size and shape. Grainy, gray, and genuinely puzzling. Under an electron microscope, its internal structure resembled membranes used by cyanobacteria for photosynthesis. On a typed manuscript draft, one careful label captured the thinking of the moment. Taken together, it was called blue-green algae in some early reports, an alga-like organism. Looking back, the algae idea was not careless. It was built from 3 real, repeatable observations. What the field still needed was to look beyond a single snapshot and watch the organism change over time. On July 18th, the FDA reported an encouraging result, a molecular signal from the recalled lettuce that looked positive. Like the final piece of a puzzle hovering just out of reach. There was still one missing piece, a direct view of the parasite itself, inside that sample. 2 specialists leaned into the same monitor, the same graph, comparing it against every control. Before anyone announced anything further, experts reviewed the signal again. The same curve resolved into a weak, irregular artifact that never crossed the real threshold. On July 19th, they walked it back. The result was not true amplification. A machine produced a signal. But a signal alone is not reliable proof. Row after row of sealed bags and an empty column where a positive result should sit. To date, there are no confirmed positive product samples from this outbreak. One red thread came off the evidence board. The correction did not erase the other evidence still sitting on the table. Interviews, the shared ingredient, purchasing and shipping records, all of it still pointed the same direction. Cases like this are not always solved with a lab sample, because by the time anyone asks, there is rarely a leftover package left to test. A shipment of lettuce gets eaten, discarded, or moved on to another kitchen long before anyone comes looking for it. And contamination may also be uneven. A single leaf, a single case, while most of a batch stays completely clean. Picture a scale, meal history, shared ingredient, supply record, together outweighing one empty lab sample. What was left were different kinds of evidence, weighed together rather than one clean answer. In practice, officials may act before waiting on final laboratory confirmation on the strength of that weight of evidence alone. The organism's own identity decades earlier was settled not by one perfect image, but by what it did next. Microscope images, tissue slides, life cycle notes, all landing on one shared table. That breakthrough was gradual and collaborative, built by researchers working in parallel, not by one single discovery. In a period incubation setup, identical samples sat labeled by the day. Researchers observed how they changed outside the body, sitting in water or soil for days at a time. Around one shared microscope, colleagues compared sample after sample. A team including Ines Ortega, a parasitologist, carried the work forward. Given time, 2 smaller compartments formed inside the mature oocyst, structures called sporocysts. Cut away the outer wall, and the structure reveals itself. And inside each of those, 2 slender forms took shape, 4 in total. The mature oocyst lined up with a clean life cycle diagram, the old algae hypothesis falling out of focus. Those internal structures were the giveaway. This was not algae. It was a cayetan. In the small intestine, those invasive forms emerge from the shell, ready to move into the body's own cells. Once inside, the parasite begins living inside cells, and the tissue responds with inflammation and damage. Researchers now understood where it matured, where it lived in the body, and what it damaged once it got there. The work came together in 2 dated milestones, 1993 for the life cycle evidence, 1994 for its formal species description. This was not one scientist shouting eureka in a single lab. It was built piece by piece, across years, across teams. The identity was settled. The next question was simpler to ask and harder to answer. Once inside a person, what exactly was it doing inside the body? Set it beside a virus and a bacterium, and the difference is immediate. Cyclospora is a one-celled parasite, not a virus or bacterium. One sporozoite meets one intestinal cell, membrane against membrane. Once inside a person, it begins entering living cells and begins to reproduce. An ordinary glass of water, a bowl of leafy greens, nothing looks out of place. The process begins the moment someone swallows it in contaminated food or water. It survives the trip protected by a tough microscopic shell, the oocyst, past the stomach down a single directional route. That shell reaches the small intestine, where it finally opens.
The small intestine's job is simple but essential. It absorbs water and nutrition from everything a person eats.
Zoom in close enough, and the surface becomes a landscape of its own.
It does that with millions of tiny finger-shaped projections called villi, lining its entire surface.
Picture the difference between a flat towel and a thick, shaggy one. One offers far more surface to work with.
A flat surface catches little, a tall, looped one catches far more.
More surface means more room to absorb what the body actually needs. At the very tips of the villi, cell by cell. This is exactly where Cyclospora enters those cells and reproduces. Where it takes hold, the tissue itself starts to change.
In response, the lining grows inflamed, its projections shorter.
What used to cross cleanly into the bloodstream now lingers instead. Shortened villi simply cannot absorb water and nutrients efficiently. 4 steps. One continuous line along the curve of the intestine. It is a clear chain. It reaches, it enters, cells are damaged. Water stays inside the gut instead of being absorbed. The result is watery diarrhea, an untouched plate, a loosened waistband. From there follow loss of appetite, weight loss, and an exhaustion that can be hard to shake. Over days, weeks, symptoms improve and return, the same relapsing pattern investigators would later recognize in 2026. Nationally, that meant 141 hospitalized. In the 5-state outbreak alone, 94 hospitalized. And the parasite still had its strangest trick after it leaves the body. When it finally leaves the body, it leaves as new protective shells, not yet ready to infect anyone. Same size, same wall, but different on the inside.
These newly released forms are immature, biologically incomplete, and harmless for the moment.
Sitting quietly in soil or water, daylight and shadow passing over them. They need about 1 to 2 weeks in the environment to finish developing. Only after that maturation are they fully formed, before they become infectious to another person. 2 people, 1 interrupted path between them. Because of that built-in delay, direct person-to-person spread is considered unlikely. That pushes investigators to look for an earlier environmental route instead: water, soil, somewhere upstream of the plate. It could start in food or water. The exact point difficult to prove, even after the fact. Laid out on a clean surface, none of it looks the least bit suspicious.
Historically, fresh produce appears repeatedly in these investigations: lettuce, raspberries, basil, cilantro.
Much of it is often eaten raw, with no cooking step to kill anything that might be hiding on it. The parasite can lodge in a leaf's own folds, creases that make it difficult to remove completely. Some of it dislodges easily. Rinsing under running water helps. But rinsing cannot guarantee that every oocyst is gone. Household disinfectants are not reliably effective against it. Heat can kill it, but that requires cooking, not just washing. Which means prevention cannot rest only on what happens at the kitchen sink, at the very end of the chain. And blaming a nationality, a region, or any group of workers along that chain is not just unfair. It is scientifically misleading. A reference manual sat open to a mostly blank page. Even after the organism finally had a name, many doctors simply did not know to look for it. That started to change in the spring of 1996, when illness clusters began appearing across multiple cities. Shipment routes converged behind an ordinary produce container. Investigators eventually linked many cases to fresh raspberries. More than 1,000 illnesses were eventually tied to that outbreak, spanning 1996 and into 1997. The organism moved in those years from specialized case reports into the broader world of food safety.
The same challenge seen in 2026 was not really new. Investigators had been here before.
In both cases, food looked fresh, parasite invisible. Nothing on the surface gave it away.
In the years between, other produce carried it too. Snow peas, cilantro, basil, and bagged salad greens. Still, that does not mean fresh produce should be feared. These events remain rare against how much is eaten safely every single day. A clinician's desk fills with overlapping possibilities. There is also a separate diagnostic problem. Symptoms resemble many other intestinal illnesses. The same watery diarrhea symptom links to several different microscopic causes. A doctor cannot identify it from diarrhea alone. One day's sample comes back empty, another sparse. It also appears unevenly in stool, which means one sample can miss it entirely. It is not part of a standard stool exam by default. Diagnosing it needs special methods, and a clinician has to specifically request the test. Laid out along one bench, 3 tools, 1 target. Those methods include specialized stains, ultraviolet light, and molecular tests.
Newer tests improve detection. That alone is not a claim that they caused the surge in cases. Treatment usually means TMP, SMX, but recognizing the illness and connecting patients to each other matters just as much as treating any one person. One anonymous interview card, alone on an otherwise empty table. Every traceback begins with individual stories, one patient at a time. The same anonymized record moves from desk to desk. The real workflow runs through 3 places in sequence: a clinician, a laboratory, a health department. Patients are asked to remember meals from days or weeks earlier, an unreasonable request that investigators make anyway, because it is the only place to start. A handful of ambiguous items, no clear pattern yet. One interview rarely solves anything on its own. Fan the cards out wide enough, and a line begins to appear through all of them. Across hundreds of interviews, a shared ingredient starts to surface. Investigators then check memories against records, receipts, invoices, delivery logs.
The traceback moves backward in 5 stages: patient, meal, restaurant, distributor, supplier.
Dozens of amber lines, all converging in one place. Across separate meals, many lines kept meeting at the same ingredient, and then at one supplier.
In the end, 2 independent paths supported the recall: interviews and supply chain records, without a positive lab sample.
The false positive correction was not a failure. It was the real process, science correcting itself, the way it should.
A blurry, fragile line becomes something solid once it passes through real verification.
That correction is exactly how confidence became deserved rather than assumed. One case map, one side sharply confirmed, the other still fading into shadow. By this point, the investigation had answered some questions and left others open. The CDC had confirmed 4,173 domestically acquired cases in 41 states as of July 20th, with 308 hospitalizations and no reported deaths. At least 7,400 additional state and local reports were not yet laboratory confirmed and still required investigation and analysis. Separately again, the 5-state outbreak counted more than 1,644 reports of its own, with 94 hospitalizations, and again, no reported deaths. Every remaining line on the board runs solid and amber, no gaps left in the chain. Interviews and supply chain records link the illness to iceberg lettuce and to a recall, enclose it on the map, and a great many other points still sit outside, unconnected. But that 5-state cluster did not explain the entire national increase. No food sample was ever confirmed positive. The precise point of contamination was not established. A microscope image, a supply chain pattern, only the verified parts passed through the frame. What remained was the discipline itself. Separate what they can see from what they can prove.
In 1991, scientists first watched this organism glowing under ultraviolet light and thought blue-green algae. In 2026, investigators traced an outbreak back to one ingredient, barely remembered by most of the people who had eaten it. The bridge between those two moments is really one continuous story. First, watching it mature under a lens. Second, following invoices and shipping records back to a source.
Between 1991 and 2026, almost everything around this parasite changed. Technology changed. The discipline did not.
One hand sorts the evidence into 3 simple stacks: confirmed, corrected, unresolved.
Follow the evidence. Correct the result when it is wrong. Act anyway, even without perfect proof. 4 objects, 1 amber thread running through all of them. What stayed visible? 1 patient, 1 microscope, 1 meal, 1 recall.
A single oocyst hangs in cinematic light, half UV blue, half warm amber.
This has been Before the Cure. If you want the next hidden history behind an outbreak, subscribe.